In Module A1, we studied G protein-coupled receptors, their structure, and how ligand binding activates G proteins through GDP-GTP exchange. In Module A2, we expanded this pathway and learned that the GS, GI, and GQ systems all begin with receptor activation, but differ in the effector enzymes they activate. These enzymes generate key second messengers, such as cyclic AMP, IP3, DAG, and calcium, which activate specific protein kinases and elicit targeted cellular responses. Together, this signaling network allows GPCRs to control a wide range of physiological functions across the body. If you have not watched Module A1 and A2, please watch the previous videos in this series for better understanding. Now, in this video, Module A3, we will explore a detailed comparison between GPCRs and ligand-gated ion channels, the major examples of each receptor family, their physiological roles, their clinical importance, and their pharmacological relevance in modern medicine. So, let's get started. Every second, trillions of cells inside the human body communicate with one another. This communication controls everything, from muscle contraction and movement to hormone secretion, emotions, memory, metabolism, and even survival itself. As discussed in the previous modules, cells communicate through specialized receptor systems that convert extracellular signals into intracellular responses through signal transduction. Among these receptor systems, two of the most important in physiology and pharmacology are G protein-coupled receptors and ligand-gated ion channels. So far, we have explored how GPCRs generate intracellular responses through signaling pathways. Now, to better understand their functional significance, let us compare GPCRs with ligand-gated ion channels and examine how differences in their signaling mechanisms lead to distinct physiological effects. Let's begin with the comparison. Although both receptor systems respond to external ligands such as neurotransmitters, hormones, and therapeutic drugs, the way they process information is fundamentally different. GPCRs elicit responses through complex intracellular signaling pathways, whereas LGICs generate immediate electrical changes by directly controlling ion movement. Understanding these differences explains how the body can perform both rapid reactions and long-lasting physiological regulation. Let us begin with their structural organization. G protein-coupled receptors, or GPCRs, belong to a class known as Metabotropic receptors. Structurally, GPCRs are composed of a single continuous protein chain that crosses the plasma membrane 7 times. Because of this characteristic arrangement, they are often called 7-transmembrane receptors, or 7-TM receptors. Unlike ion channels, GPCRs do not contain a pore through which substances can pass. Instead, they function as molecular switches. When a ligand binds to the receptor on the extracellular side of the membrane, the receptor undergoes a 3-dimensional conformational change. This structural rearrangement creates an intracellular docking surface that interacts with a heterotrimeric G protein composed of alpha, beta, and gamma subunits. Once activated, the G protein separates and begins transmitting signals inside the cell. Rather than directly producing electrical changes, GPCR activation modifies intracellular chemistry and cellular metabolism through signaling pathways. That is why these receptors are classified as metabotropic receptors. Now compare this mechanism with ligand-gated ion channels. Ligand-gated ion channels belong to a different functional category known as ionotropic receptors. Unlike GPCRs, these receptors are not formed from a single protein chain. Instead, they consist of multiple protein subunits, typically 4 or 5, arranged together in a circular structure that forms a central water-filled pore across the plasma membrane. This pore acts as a physical gate. When an appropriate ligand binds to the receptor, the protein subunits undergo a conformational change that rapidly opens or closes the channel. Once open, ions move directly across the membrane following their electrochemical gradient. Because these receptors directly regulate ion movement and alter membrane potential, they are classified as ionotropic receptors. This structural distinction creates two completely different methods of signaling. GPCR signaling is indirect and highly amplified. The process begins when a ligand binds to the receptor. The activated receptor then stimulates the associated G protein by promoting exchange of GDP for GTP. The activated G protein subsequently interacts with intracellular effector enzymes such as adenylyl cyclase or phospholipase C. These enzymes generate intracellular second messengers, including cyclic AMP, IP3, DAG, and calcium ions. These signaling molecules spread throughout the cell and activate multiple downstream pathways, ultimately producing the physiological response. Because the signal must pass through several intracellular steps, GPCR signaling is not immediate. However, this complexity provides an important advantage. Signal amplification. A single receptor can activate many G proteins. Each G protein can regulate enzymes, and each enzyme can generate thousands of second messenger molecules. As a result, even very small concentrations of hormones can produce powerful biological effects. In contrast, ligand-gated ion channels operate in a much simpler manner. Once a ligand binds, the channel opens immediately. Ions move across the membrane. Membrane potential changes, and the cell responds. No G proteins are involved, no enzymes are required, and no second messengers are generated. Typically, one activated receptor produces one channel response. Therefore, signal amplification is minimal, and the effects are usually localized and limited. Let us now compare the speed and physiological roles of these receptor systems. GPCRs operate on a relatively slow timescale. Their responses typically develop over seconds to minutes because multiple intracellular signaling steps are required. However, this slower signaling provides greater regulation and modulation. As a result, GPCRs play important roles in hormonal signaling, heart rate regulation, and metabolic control. In contrast, ligand-gated ion channels act extremely rapidly. Their responses occur within milliseconds because ions flow directly through the channel. This fast signaling is essential for rapid electrical communication. Consequently, ionotropic receptors mediate functions such as reflex actions, skeletal muscle contraction, and fast synaptic transmission. Thus, GPCRs are specialized for regulation and long-term control, whereas ligand-gated ion channels are specialized for immediate responses. Another important difference is the duration of the response. GPCR-mediated effects are generally long-lasting. Even after the ligand is removed, intracellular signaling molecules and activated enzymes may continue to influence cellular activity. Some pathways can even activate transcription factors and alter gene expression. As a result, GPCR effects may persist for hours or even days. In contrast, responses mediated by ligand-gated ion channels are brief and transient. Once the channel closes, signaling ends rapidly. These receptors do not activate gene transcription and are specialized for immediate responses. Several important ionotropic receptors illustrate these functions. The nicotinic acetylcholine receptor mediates neuromuscular transmission and skeletal muscle contraction. The GABA-A receptor is the major inhibitory receptor in the central nervous system. The glycine receptor performs a similar inhibitory role in the spinal cord. And the 5-HT3 receptor participates in rapid vomiting reflex pathways. Examples of GPCRs further illustrate their role in long-term physiological regulation. Adrenergic receptors respond to catecholamines and regulate cardiovascular and metabolic functions. Muscarinic receptors mediate parasympathetic responses, such as slowing heart rate and stimulating glandular secretion. Dopamine receptors influence movement, reward pathways, and endocrine regulation. Histamine receptors participate in allergic responses and regulate gastric acid secretion. Taken together, these examples highlight the primary specialization of GPCRs. They are designed for amplified and sustained signaling. In contrast, ligand-gated ion channels are specialized for rapid electrical signaling. Therefore, GPCRs primarily provide long-term physiological regulation, whereas ligand-gated ion channels mediate immediate physiological responses. Together, these two receptor systems enable the body to combine rapid reactions with precise long-term control, ensuring efficient cellular communication and physiological homeostasis. Many clinically important receptors belong to the G-protein-coupled receptor family. Although they share a common mechanism of activation, different receptor subtypes couple to different G proteins and elicit distinct physiological responses. Let us explore some of the major GPCR families. We will begin with one of the most clinically important groups, the adrenergic receptors. These receptors are activated by the catecholamines epinephrine and norepinephrine and mediate the physiological effects of the sympathetic nervous system. They are classified into alpha and beta receptor subtypes, each coupled to specific G proteins and producing distinct responses. We begin with the alpha-1 receptor. Alpha-1 receptors are coupled to GQ proteins and are located primarily in vascular smooth muscle, the eye, and bladder sphincters. Activation of these receptors stimulates the phospholipase C pathway, leading to vasoconstriction, pupil dilation, or mydriasis, and increased sphincter tone. Because excessive alpha-1 activity contributes to elevated blood pressure, alpha-1 blockers such as prazosin and doxazosin are commonly used in the treatment of hypertension. Next are the alpha-2 receptors. These receptors are coupled to GI proteins and are located mainly on presynaptic sympathetic nerve terminals. Their activation inhibits adenylyl cyclase and decreases the release of norepinephrine. This negative feedback mechanism reduces sympathetic outflow. A clinically important drug acting on alpha-2 receptors is clonidine, an agonist used in the management of hypertension. Moving to the beta receptors, beta-1 receptors are coupled to G's proteins and are found predominantly in the heart and in the juxtaglomerular cells of the kidney. Activation of beta-1 receptors increases heart rate, enhances the force of cardiac contraction, and stimulates renin release from the kidney. Because excessive beta-1 stimulation can be harmful in cardiovascular disease, selective beta blockers such as metoprolol and atenolol are widely used, particularly in heart failure and other cardiac disorders. Beta-2 receptors are also coupled to G's proteins. They are present in bronchial smooth muscle, vascular smooth muscle, the uterus, and skeletal muscle. Activation of beta-2 receptors causes relaxation of smooth muscle, resulting in bronchodilation and improved airflow. This property makes beta-2 agonists, such as salbutamol, highly effective in the treatment of asthma and other obstructive airway diseases. Finally, beta-3 receptors are likewise coupled to G's proteins. They are expressed predominantly in adipose tissue, where they regulate lipolysis and fat metabolism. However, from a clinical perspective, these receptors are also functionally expressed in the detrusor muscle of the bladder wall. Activation of beta-3 receptors relaxes the detrusor muscle and increases bladder capacity. A selective beta-3 agonist, mirabegron, is therefore used in the treatment of overactive bladder. Let us now turn our attention to the muscarinic receptors, another important family of G-protein-coupled receptors. These receptors are activated by acetylcholine and mediate many of the physiological effects of the parasympathetic nervous system, often summarized as the rest and digest responses. They are classified into 5 subtypes, M1 through M5. Through different signaling pathways, muscarinic receptors regulate a wide range of functions, including cognition, cardiac activity, smooth muscle contraction, and glandular secretion. We begin with the M1 receptor. M1 receptors are coupled to GQ proteins and are found mainly in the central nervous system and gastric parietal cells. Activation of these receptors stimulates the phospholipase C pathway, leading to increased intracellular calcium levels. Functionally, M1 receptors play an important role in cognitive processing and memory, and they also stimulate gastric acid secretion. From a pharmacological standpoint, drugs such as pirenzepine, an M1 selective antagonist, have been used to reduce gastric acid secretion, while acetylcholinesterase inhibitors, such as donepezil, indirectly enhance M1-mediated signaling and are used in the treatment of Alzheimer's disease. Next is the M2 receptor. M2 receptors are coupled to GI proteins and are located predominantly in the heart, especially in the sinoatrial and atrioventricular nodes. Activation of M2 receptors inhibits adenylyl cyclase and decreases cyclic AMP levels. As a result, parasympathetic stimulation slows the heart by reducing the rate of impulse generation and conduction. Thus, M2 receptors are responsible for the characteristic effect of acetylcholine in producing a decrease in heart rate. Clinically, blockade of these receptors by atropine, a muscarinic antagonist, increases heart rate and is widely used to manage symptomatic bradycardia and atrioventricular block. Next, we have the M3 receptor. Like M1 receptors, M3 receptors are coupled to GQ proteins and activate the phospholipase C pathway. These receptors are widely distributed in smooth muscle and exocrine glands. Their activation produces bronchoconstriction, increases glandular secretion, and causes smooth muscle contraction in organs such as the gastrointestinal tract, urinary bladder, and eye. From a clinical perspective, M3 receptors are particularly important in respiratory diseases. Drugs such as ipratropium and tiotropium, which act as muscarinic receptor antagonists, block M3-mediated bronchoconstriction and promote bronchodilation. Therefore, they are widely used in the treatment of chronic obstructive pulmonary disease, or COPD. Similarly, oxybutynin, another M3 antagonist, relaxes the urinary bladder and is used in patients with an overactive bladder. Moving to the M4 receptor, this receptor is coupled to GI proteins and is found predominantly in the central nervous system, particularly in the striatum and cerebral cortex. Activation of M4 receptors inhibits adenylyl cyclase and decreases cyclic AMP levels. Functionally, these receptors modulate neurotransmitter release and play an important role in regulating dopaminergic signaling. Although few highly selective drugs target M4 receptors, they have emerged as promising therapeutic targets in neuropsychiatric disorders. More recently, xanomeline trospium, a muscarinic agonist combination, has demonstrated therapeutic benefit in schizophrenia through preferential activation of M1 and M4 receptors. In addition, M4 receptors continue to be investigated as potential targets in Parkinson's disease and other central nervous system disorders. Finally, we have the M5 receptor. M5 receptors are coupled to GQ proteins and are expressed mainly in specific regions of the brain and in cerebral blood vessels. Activation of M5 receptors stimulates the phospholipase C pathway, contributing to dopamine release and cerebral vasodilation. Because of their involvement in reward pathways and cerebral blood flow, M5 receptors are being studied in relation to drug addiction and neuropsychiatric disorders. At present, no selective drugs targeting M5 receptors are available, and much of their physiological and pharmacological significance remains an active area of research. In fact, muscarinic receptors mediate many of the physiological effects of the parasympathetic nervous system. Among them, M1, M2, and M3 receptors are the most clinically important, whereas M4 and M5 receptors are primarily involved in central nervous system functions and remain promising targets for future therapeutic development. Let's now focus on the dopamine receptors, another important family of G-protein-coupled receptors. These receptors are activated by dopamine and play essential roles in motor control, cognition, endocrine regulation, and reward pathways. Dopamine receptors are broadly divided into two groups: the D1-like receptors, consisting of D1 and D5, which are coupled to GS proteins, and the D2-like receptors, comprising D2, D3, and D4 receptors, which are coupled to GI proteins. We begin with the D1 receptor. D1 receptors are coupled to GS proteins and are found in the kidney, vascular smooth muscle, and several regions of the brain. Activation of these receptors stimulates adenylyl cyclase and increases cyclic AMP levels. One of their most important physiological actions is renal vasodilation, which helps maintain renal blood flow. In the central nervous system, D1 receptors also contribute to motor control and cognitive functions. From a pharmacological standpoint, dopamine agonists such as phenoldiprem, a selective D1 receptor agonist, produce vasodilation and are used intravenously in the management of severe hypertension and hypertensive emergencies. Moving to the D2 receptor. These receptors are coupled to GI proteins and are located in the basal ganglia, pituitary gland, and presynaptic nerve terminals. Their activation inhibits adenylyl cyclase and decreases cyclic AMP levels. Functionally, D2 receptors regulate movement, inhibit prolactin secretion, and reduce neurotransmitter release. From a clinical standpoint, D2 receptors are particularly important because they are the primary targets of many antipsychotic drugs. Agents such as haloperidol, risperidone, and olanzapine block D2 receptors and are widely used in the treatment of schizophrenia and other psychotic disorders. Conversely, D2 agonists, such as bromocriptine and cabergoline, suppress prolactin secretion and are used to treat hyperprolactinemia and prolactin-secreting pituitary adenomas. Beyond D1 and D2 receptors, 3 additional dopamine receptor subtypes have been identified. The D3 receptor, which is also coupled to GI proteins, is located mainly in the limbic system and plays a role in emotional behavior, motivation, and reward pathways. Because of these functions, D3 receptors are being investigated as potential targets in addiction and psychiatric disorders. Interestingly, dopamine agonists such as pramipexole and ropinirole, which are used to treat Parkinson's disease and restless leg syndrome, exhibit significant affinity for D3 receptors, contributing to their therapeutic effects. Similarly, the D4 receptor belongs to the D2-like family and is coupled to GI proteins. It is expressed predominantly in the frontal cortex and limbic regions of the brain, where it contributes to cognition, attention, and emotional processing. Certain atypical antipsychotic drugs, particularly clozapine, exhibit relatively high affinity for D4 receptors, although the precise clinical significance of this interaction remains under investigation. Other atypical antipsychotics, including quetiapine, also interact with D4 receptors to varying degrees. Finally, the D5 receptor belongs to the D1-like family and is coupled to GS proteins. It is found mainly in the hippocampus and hypothalamus. Activation of D5 receptors stimulates adenylyl cyclase and contributes to learning, memory, and neuroendocrine regulation. At present, no drugs selectively target D5 receptors, and much of their physiological significance remains an active area of research. Before we conclude, it is important to note that due to the extensive scope of this module, the remaining topics will be covered separately in the second part of Module A3. In the second part of Module A3, we will examine the remaining receptor families, including the histamine, serotonin, opioid, and angiotensin receptor families. We will then integrate these concepts by discussing their physiological functions, clinical importance, and pharmacological relevance. Together, these topics will provide a comprehensive understanding of the clinical significance of G-protein-coupled receptors. Thank you for joining me, and I look forward to seeing you in the next part of Module A3.